Hello and welcome. In order to better understand how to target metabolic pathways in cancer cells, let's examine their characteristics and the ways they are different from normal cells. Cancer cells exhibit several key characteristics that distinguish them from their normal counterparts. These characteristics, often referred to as the hallmarks of cancer, provide insights into the complexities of this disease and guide the development of innovative treatment strategies. Doctors Douglas Hanahan and Robert A. Weinberg, in their seminal articles published in Cell in 2000 and in 2011, outlined these hallmarks, providing a framework for understanding the intricate mechanisms underlying cancer development and progression. In this video, we will delve into the 10 hallmarks of cancer, exploring each hallmark in detail and discussing its implications for cancer therapy. For those less technically oriented, I recommend the Scientific American series, which offers accessible explanations of complex scientific concepts for further reading.
The first hallmark of cancer is self-sufficiency in growth signals. Normal cells require external signals such as growth factors to initiate and regulate cell division. These signals bind to specific receptors on the cell surface, triggering a cascade of intracellular events that ultimately lead to cell proliferation. Cancer cells, however, develop the ability to produce their own growth signals or hijack the signaling pathways of surrounding cells, effectively bypassing the need for external stimuli. This self-sufficiency in growth signals allows cancer cells to proliferate uncontrollably, leading to tumor formation. Imagine a car that can accelerate without a driver pressing the gas pedal. That's essentially what's happening with cancer cells. They have acquired the ability to hit the gas on their own, driving their uncontrolled growth. This hallmark is often associated with mutations in genes encoding growth factor receptors, or downstream signaling molecules. For example, some cancers overexpress epidermal growth factor receptor, or EGFR, a receptor that plays a crucial role in cell growth and survival. When EGFR is overexpressed, it sends continuous growth signals to the cancer cell, even in the absence of external growth factors. Understanding the mechanisms underlying self-sufficiency in growth signals is crucial for developing targeted therapies that can block these aberrant signaling pathways and inhibit cancer cell proliferation. By targeting these specific pathways, we can potentially cut the brakes on cancer cell growth. For instance, EGFR inhibitors such as cetuximab and erlotinib have shown promise in treating certain types of cancer, particularly those with EGFR overexpression. These drugs work by binding to EGFR and blocking its activation, thereby inhibiting the downstream signaling cascade that drives cancer cell growth. However, cancer cells are cunning adversaries, and they often find ways to evade these targeted therapies. Resistance to EGFR inhibitors is a growing concern, highlighting the need for continued research and development of novel therapeutic strategies.
The second hallmark of cancer is insensitivity to anti-growth signals. In normal tissues, cell growth is tightly regulated by a balance of pro-growth and anti-growth signals. Anti-growth signals, also known as growth inhibitors, act as brakes on cell proliferation, preventing excessive growth and maintaining tissue homeostasis. Cancer cells, however, develop the ability to ignore these anti-growth signals, allowing them to proliferate unchecked, much like a car that can't stop because its brakes are broken. This insensitivity to anti-growth signals is often associated with mutations in genes encoding tumor suppressor genes, such as p53 and retinoblastoma, or RB. Tumor suppressor genes act as guardians of the genome, protecting cells from uncontrolled growth and DNA damage. They play critical roles in regulating cell cycle checkpoints, inducing apoptosis, or programmed cell death, in response to DNA damage, and inhibiting cell proliferation in response to anti-growth signals. When these tumor suppressor genes are mutated or inactivated, the brakes on cell growth are released, allowing cancer cells to proliferate unchecked. It's like having a broken traffic light. Without the red light to stop them, cancer cells keep dividing and growing. For example, p53, often referred to as the guardian of the genome, is a crucial tumor suppressor gene that regulates cell cycle arrest, DNA repair, and apoptosis. Mutations in p53 are found in over 50% of human cancers, highlighting its central role in tumorigenesis. Similarly, RB, another key tumor suppressor gene, regulates the transition from the G1 to S phase of the cell cycle, a critical checkpoint that controls cell division. Mutations in RB disrupt this checkpoint, allowing cells to bypass normal growth control mechanisms and proliferate uncontrollably. Understanding the mechanisms underlying insensitivity to anti-growth signals is crucial for developing targeted therapies that can restore the function of these critical tumor suppressor pathways. By reactivating these pathways, we can potentially fix the brakes on cancer cell growth.
The third hallmark of cancer is the evasion of apoptosis. Apoptosis, also known as programmed cell death, is a fundamental biological process that plays a critical role in maintaining tissue homeostasis and eliminating damaged or unwanted cells. It's like a cellular suicide program that gets activated when cells are no longer needed or become damaged. In normal cells, apoptosis is tightly regulated by a complex interplay of pro-apoptotic and anti-apoptotic proteins. When cells are stressed, damaged, or no longer needed, pro-apoptotic proteins are activated, initiating a cascade of events that culminate in cell death. Cancer cells, however, develop mechanisms to evade apoptosis, allowing them to survive and proliferate even when they're damaged or no longer needed. This evasion of apoptosis is akin to disabling the self-destruct button, rendering cancer cells immortal and resistant to cell death. One way cancer cells evade apoptosis is by upregulating anti-apoptotic proteins such as BCL2. BCL2 family proteins play a pivotal role in regulating apoptosis by controlling the release of cytochrome C from mitochondria, a key event in the apoptotic pathway. Overexpression of BCL2, as seen in various cancers, can tip the balance towards cell survival, inhibiting apoptosis and promoting cancer cell survival. This is like having a bodyguard for the self-destruct button, preventing it from being pressed. Another mechanism of apoptosis evasion involves mutations in the p53 gene, which we discussed earlier. Remember that p53 is a tumor suppressor gene that plays a crucial role in inducing apoptosis in response to DNA damage. When p53 is mutated or inactivated, cells with damaged DNA are no longer efficiently eliminated, increasing the risk of accumulating mutations and progressing towards cancer. It's like having a faulty alarm system that fails to alert the authorities when there's an intruder. Understanding the mechanisms underlying apoptosis evasion is crucial for developing targeted therapies that can restore the apoptotic pathway and induce cancer cell death. By reactivating the self-destruct button, we can eliminate cancer cells and prevent tumor growth.
The fourth hallmark of cancer is replicative immortality. Normal cells have a finite lifespan and can only divide a limited number of times before entering a state of senescence, where they cease to divide. This limited replicative potential is due to the gradual shortening of telomeres, protective caps at the ends of chromosomes that shorten with each cell division. Telomeres act like the plastic tips on shoelaces, preventing chromosomes from fraying and sticking together. However, with each cell division, telomeres shorten, and once they reach a critically short length, cells can no longer divide and enter senescence. Think of it like a clock that ticks down with each cell division. Cancer cells, however, have found a way to turn back the clock, achieving replicative immortality by upregulating the enzyme telomerase. Telomerase is an enzyme that adds nucleotides to telomeres, replenishing their length and allowing cancer cells to divide indefinitely. It's like having a magical cobbler who can repair the worn-out tips of your shoelaces, allowing you to keep walking forever. This ability to divide indefinitely is a key characteristic of cancer cells, enabling them to form tumors and invade other tissues. It's like having a car that can drive forever without running out of gas. The discovery of telomerase and its role in cancer has opened up new avenues for therapeutic intervention. Telomerase inhibitors, drugs that block telomerase activity, are being investigated as potential cancer therapies. By inhibiting telomerase, we can potentially limit the replicative potential of cancer cells and halt their uncontrolled growth. It's like removing the magical cobbler, preventing the shoelaces from being repaired. However, telomerase is also expressed in some normal cells, such as stem cells, which need to divide continuously. Therefore, developing telomerase inhibitors that specifically target cancer cells without harming normal cells is a major challenge.
The fifth hallmark of cancer is the induction of angiogenesis, also known as tumor vascularity. As tumors grow larger, they require a constant supply of oxygen and nutrients to survive and proliferate. To meet this demand, cancer cells stimulate the formation of new blood vessels. a process called angiogenesis. Angiogenesis is a normal physiological process that is essential for wound healing, tissue repair, and embryonic development. However, in cancer, angiogenesis is hijacked by tumor cells to support their uncontrolled growth and spread. It's like building new roads to supply a growing city. Cancer cells release pro-angiogenic factors, such as vascular endothelial growth factor, VEGF, which stimulate the growth of new blood vessels from pre-existing ones. VEGF binds to receptors on endothelial cells, the cells that line blood vessels, triggering a signaling cascade that promotes cell proliferation, migration, and the formation of new blood vessels. This increased blood supply provides tumors with the oxygen and nutrients they need to grow and metastasize. It also provides a route for cancer cells to escape from the primary tumor and spread to other parts of the body. Imagine a river that branches out, allowing boats, cancer cells, to travel to different destinations, other organs. Targeting angiogenesis has emerged as a promising strategy for cancer therapy. Anti-angiogenic therapies, such as VEGF inhibitors, aim to block the formation of new blood vessels, depriving tumors of the nutrients and oxygen they need to grow. These therapies essentially cut off the supply lines to the tumor, starving it and preventing its growth. Several anti-angiogenic drugs have been approved for the treatment of various cancers, including bevacizumab, sunitinib, and sorafenib. These drugs have shown promise in extending survival and improving quality of life for cancer patients. However, resistance to anti-angiogenic therapies is a growing concern, highlighting the need for continued research and development of novel therapeutic strategies.
The sixth hallmark of cancer is invasion and metastasis, the process by which cancer cells spread from the primary tumor to distant sites in the body. This is a complex multi-step process that involves several steps. Cancer cells first invade the surrounding tissue, breaking free from the primary tumor and infiltrating nearby tissues. Imagine a plant whose roots start to grow into the surrounding soil. Cancer cells then penetrate the walls of blood vessels or lymphatic vessels, entering the circulatory system. This is like the plant's roots reaching a water source and entering the flow. Once in the circulation, cancer cells travel through the bloodstream or lymphatic system to distant sites in the body. This is like the plant's seeds being carried by the water to different locations. Cancer cells then exit the blood vessels or lymphatic vessels at distant sites, entering the surrounding tissue. This is like the plant's seeds settling in new soil. Finally, cancer cells establish secondary tumors called metastases at these distant sites. This is like the plant seeds germinating and growing into new plants. Metastasis is a major cause of cancer mortality, as it makes the disease much more difficult to treat. Several factors contribute to the invasive and metastatic potential of cancer cells, including cancer cells lose the adhesion molecules that keep normal cells together, allowing them to break free from the primary tumor and invade surrounding tissues. Cancer cells acquire increased motility, allowing them to migrate through tissues and enter blood vessels or lymphatic vessels. Cancer cells secrete enzymes that break down the extracellular matrix, the scaffolding that surrounds cells, creating pathways for invasion. Understanding the mechanisms underlying invasion and metastasis is crucial for developing therapies that can prevent or inhibit this process. Researchers are investigating various approaches, including drugs that inhibit the function of cell-cell adhesion molecules, preventing cancer cells from breaking free from the primary tumor. Therapies that block the signaling pathways that promote cancer cell migration. Drugs that inhibit the activity of matrix-degrading enzymes, preventing cancer cells from creating pathways for invasion.
The seventh hallmark of cancer is genome instability. Cancer cells are characterized by a high degree of genetic instability, meaning that their DNA is more prone to mutations than normal cells. This instability arises from defects in DNA repair mechanisms, leading to an accumulation of genetic alterations over time. Imagine a game of telephone where the message gets distorted with each retelling. In cancer cells, DNA replication is like that. Game errors creep in with each cell division, leading to an accumulation of mutations. These mutations can affect a wide range of genes, including genes that promote cell growth and proliferation. Mutations in oncogenes can activate them, leading to uncontrolled cell growth. Genes that inhibit cell growth and promote apoptosis. Mutations in tumor suppressor genes can inactivate them, removing the brakes on cell growth. genes involved in repairing DNA damage. Mutations in DNA repair genes can further increase the rate of mutations, creating a vicious cycle of genomic instability. This genomic instability is a double-edged sword for cancer cells. On the one hand, it fuels tumor heterogeneity, generating a diverse population of cancer cells with different characteristics. This diversity makes it more likely that some cancer cells will survive and thrive, even in the face of chemotherapy or other treatments. On the other hand, genomic instability also makes cancer cells more vulnerable to certain therapies, such as those that induce DNA damage. For example, radiation therapy and some chemotherapies work by damaging DNA, which can kill cancer cells or prevent them from dividing. Understanding the mechanisms underlying genome instability is crucial for developing therapies that can exploit this vulnerability. Researchers are investigating various approaches, including drugs that inhibit specific DNA repair pathways, making cancer cells more sensitive to DNA-damaging agents. Identifying combinations of mutations that make cancer cells particularly vulnerable to specific drugs.
The eighth hallmark of cancer is the upregulation of inflammation. Inflammation is a complex biological process that is essential for wound healing, tissue repair, and defense against infection. It's like a fire alarm in the body, alerting the immune system to danger. However, chronic inflammation can contribute to the development and progression of cancer. Imagine a fire alarm that keeps going off, even when there's no fire. This constant state of alert can damage tissues and create an environment that favors tumor growth. Cancer cells can directly and indirectly promote inflammation, creating a vicious cycle that fuels tumor development. Directly, cancer cells release inflammatory mediators, such as cytokines and chemokines, which recruit immune cells to the tumor microenvironment. Indirectly, cancer cells can induce inflammation by damaging surrounding tissues or by activating inflammatory pathways in nearby cells. This inflammatory microenvironment can promote tumor growth in several ways, stimulating angiogenesis. Inflammatory cells release growth factors that promote angiogenesis, providing tumors with the nutrients and oxygen they need to grow, suppressing the immune system. Chronic inflammation can suppress the immune system, creating an environment where cancer cells can evade immune destruction, promoting tumor cell proliferation and survival. Inflammatory mediators can directly stimulate tumor cell proliferation and protect them from apoptosis. Targeting inflammation has emerged as a promising strategy for cancer prevention and treatment. Several anti-inflammatory drugs such as aspirin, celecoxib, and infliximab are being investigated for their potential to reduce cancer risk or slow tumor growth. However, inflammation is a complex process with both beneficial and detrimental effects. Therefore, developing anti-inflammatory therapies that specifically target tumor-promoting inflammation without compromising the immune system's ability to fight infection is a major challenge. The ninth hallmark of cancer is the reprogramming of energy metabolism. Cancer cells have a voracious appetite for energy, as they need to fuel their rapid growth and proliferation. To meet this demand, cancer cells rewire their metabolism, shifting from oxidative phosphorylation, the efficient energy-producing pathway used by normal cells, to glycolysis, a less efficient pathway that produces energy even in the absence of oxygen. This metabolic shift, known as the Warburg effect, allows cancer cells to thrive in the hypoxic, or low oxygen conditions often found within tumors. Imagine a car that can switch from gasoline to diesel fuel, allowing it to run on different types of energy. The Warburg effect provides cancer cells with several advantages. Rapid energy production. Glycolysis produces energy, or ATP, much faster than oxidative phosphorylation, although it is less efficient overall. Biosynthetic precursors. Glycolysis provides cancer cells with the building blocks they need to synthesize new proteins, lipids, and nucleic acids, essential for rapid growth. Resistance to oxidative stress. Glycolysis reduces the production of reactive oxygen species, or ROS, which can damage DNA and other cellular components. Targeting cancer metabolism has emerged as a promising therapeutic strategy. Several drugs that target metabolic pathways are being investigated for their potential to selectively kill cancer cells or inhibit their growth. These approaches include inhibiting glycolysis, drugs that block key enzymes involved in glycolysis, depriving cancer cells of their energy source; activating oxidative phosphorylation, drugs that stimulate oxidative phosphorylation, forcing cancer cells to rely on a less favorable metabolic pathway; Targeting metabolic dependencies, identifying specific metabolic vulnerabilities in different types of cancer and developing drugs that exploit these vulnerabilities.
The 10th and final hallmark of cancer is the evasion of destruction by the host immune system. The immune system is our body's natural defense against disease, constantly patrolling for and eliminating foreign invaders, such as bacteria, viruses, and even cancer cells. However, cancer cells are masters of disguise, evolving sophisticated mechanisms to evade immune detection and destruction. Imagine a criminal wearing a disguise to avoid being caught by the police. Cancer cells employ several strategies to evade the immune system, downregulating tumor antigens. Cancer cells can reduce the expression of tumor antigens, molecules that mark them as different from normal cells, making it harder for the immune system to recognize them. Secreting immunosuppressive molecules. Cancer cells release molecules that suppress the immune response, creating an immunosuppressive microenvironment that protects them from immune attack. Inducing immune cell exhaustion. Chronic exposure to tumor antigens can exhaust immune cells, rendering them less effective at killing cancer cells. Immunotherapy, a revolutionary approach to cancer treatment, aims to harness the power of the immune system to fight cancer. Several immunotherapy drugs have been approved for the treatment of various cancers, including immune checkpoint inhibitors. Drugs that block immune checkpoints, molecules that normally keep the immune response in check, unleashing the immune system to attack cancer cells. Adoptive cell transfer, a personalized therapy that involves isolating and expanding a patient's own immune cells in the laboratory and then infusing them back into the patient to boost their immune response against cancer. Cancer vaccines, vaccines that stimulate the immune system to recognize and attack cancer cells. Immunotherapy has shown remarkable success in treating some cancers, leading to long-lasting remissions and even cures. However, not all patients respond to immunotherapy, and some experience serious side effects. Therefore, further research is needed to improve the efficacy and safety of immunotherapy and to identify biomarkers that can predict which patients are most likely to benefit from this treatment.
In this video, we explored the 10 hallmarks of cancer, a framework for understanding the complex mechanisms underlying cancer development and progression. These hallmarks highlight the remarkable adaptability of cancer cells, their ability to overcome numerous obstacles to survive, proliferate, and spread. Understanding these hallmarks is crucial for developing targeted therapies that can effectively combat cancer by disrupting these unique characteristics. By exploiting the vulnerabilities revealed by these hallmarks, we can develop more effective and less toxic therapies that target cancer cells while sparing normal cells. For those seeking a more in-depth understanding of the hallmarks of cancer, I encourage you to read the seminal articles by Drs. Hanahan and Weinberg and explore the Scientific American series for accessible explanations of complex scientific concepts.